Welcome, everybody, to the latest episode of Star Cells and God. This is the show where we discuss new discoveries taking place at the frontiers of science and describe how these discoveries provide evidence for God's existence, God's character, and the reliability of the Old and the New Testaments. My name is Fuz Rana. I'm a biochemist and a Christian apologist, and I work for an organization called Reasons to Believe, which is the organization that sponsors Star Cells and God. If you want to know more about Reasons to Believe, I invite you to go to our website, www.reasons.org. Also, you can follow us on social media, RTB_official. And then last but not least, please go to our YouTube channel, Reasons to Believe, and subscribe. Also, make sure you use the notification button so that you are alerted the next time an episode of Star Cells and God drops. Okay, well, without any further ado, let's go ahead and get started. Hugh, I forgot to introduce you. I'm sorry. I'm joined in studio by Hugh Ross, who is an astronomer, an astrophysicist, the founder of Reasons to Believe. And you're going to be talking today about time dilation in the universe. So hopefully this won't take too long. That's my really bad joke. And then I'm going to be talking about the design of protein machines. So anyway, Hugh, why don't you go ahead and get us started?
Well, let me start off with a quiz for you. Okay, here's a quiz. What is the one equation that everybody in the world who's gotten past the third grade knows?
It's probably E equals MC squared.
You're right. I mean, I always thought it'd be something like F equals MA, and I use that, people have a blank stare in their face. But E equals MC squared, they know.
It's not the quadratic equation. Nobody remember. Well, I've always told in algebra, memorize this because you'll always use it.
Well, you will, but E equals MC squared is the only equation I can count on people knowing.
Yeah.
And people credit Albert Einstein with the brilliance for developing that equation. It's actually something that could be derived by any high school student who's got basic algebra under their belt. Mm-hmm. Where Einstein deserves credit is for his theory of general relativity. Mm-hmm. There you need to be fluent with tensor calculus to develop that. But E equals MC squared, it's quite straightforward. It's easy to derive just from basic principles of physics. And I do have an equation I'm going to show you here. So we can pull up this slide on the screen. This equation you can derive from E equals MC squared. I'm not going to show you the derivation. I thought it'd be fun to leave that for the audience.
Yeah.
Starting with E equals MC squared, you can come up with this equation. And basically, this tells us that if Einstein's right about special relativity, distant clocks in the universe will run slower. That's because we live in a Big Bang universe. And in a Big Bang universe, the universe is expanding. And with a general expansion of the universe, the farther away a galaxy or a quasar is, the faster it'll appear to be moving away from us. And the faster it moves away, the slower clocks on that galaxy or quasar will run. I mean, here's a thought experiment. We could actually send you to the Andromeda galaxy at 0.999% the velocity of light. It would take you less than a year to get there. You might want to spend a year touring around there and come back. So you would come back a few years older. But if I wait for you to come back, clocks run slower for you than they do for me, I would be 5 million years older because it's 2.5 million light years. I got to wait 5 million years. But you come back, you're only a couple of years older. You look great. Where's Hugh? He's been dead for 5 million years. So that's kind of an example of what happens with time dilation. Mm-hmm. And it's this factor here. The clocks will run slower by a factor of 1 divided by the square root of 1 minus v squared divided by c squared. I decided to say it aloud because I know not everybody is going to be watching this recording on video. So for audio, that's it. And v is the velocity of the galaxy relative to the Earth. Mm-hmm. So for example, that's how fast away a galaxy is moving relative to us. C is the velocity of light. Now, the effect that this has, we can pull up the next slide. It basically tells you, okay, if we're looking far away, and we're talking about looking like 8 billion light years away, the expansion of the universe means that that object, relative to us, will be moving at about half the velocity of light. So if it's moving at half the velocity of light relative to us, the clocks in that galaxy are going to run about 15% slower. Or pardon me, this is for 0.25. So 1/4 of the velocity of light, which is like about 6-7 billion light years away, it's going to run 15% slower. But because of that square factor, if you go to, say, 0.9 c, 9/10 of velocity of light, which is what you're going to see if you're within the first billion years of the history of the universe, so galaxies that are 13 billion light years away, they're going to be moving relative to us at more than 90% the velocity of light. Now the time dilation factor is really substantial. That clock is going to run 526% slower. So it's going to be like 5 times slower or more than clocks here on Earth. Now, here's a problem that astronomers have faced. I mean, this is a major prediction of the Big Bang creation model. And the reason why I'm focusing on this, the Bible thousands of years ago explicitly stated repeatedly at least 3, if not 4, of the fundamental features of what we call the Big Bang model. And that's why when astronomers first discovered that the universe had Big Bang features, they said, we got to get rid of this. This is what the Bible says. And so those astronomers that weren't happy with Christianity or the biblical message for a 50-year period tried to find some way to get rid of the Big Bang. And so inherent in astronomy is the recognition. Big Bang cosmology establishes the inerrancy of the Bible and the credibility of the Christian faith. But a major prediction of Big Bang cosmology is that distant clocks will run slower, and they're going to run slower at precisely these predicted rates. And so one way to test the Big Bang is to look at clocks in distant galaxies and quasars and see if they actually run slower by the precise amount predicted by the Big Bang creation model. So, and astronomers have been working on this for 30 years. The problem is the clocks that we're most familiar with, you can't see them in galaxies far away. I mean, the best known clock is what's called a Cepheid variable star. These are bright stars, and the period with which they go through their oscillation, if it's a 40-day light period, that means it's much brighter than if it's a 3-day light period. And so they measure the period of the variation. They know how bright it is. But with that time dilation, those periods are going to become longer.
Okay.
But the problem is we can only see Cepheid variable stars in galaxies that are within, say, 40 million light years of us.
Okay.
And at 40 million light years away, the time dilation effect is essentially zero.
Right.
The galaxies are not moving fast enough relative to the velocity of light to make a difference. And so we really need to find clocks in galaxies that are billions of light years away. And the one candidate is what we call a type 1a supernova eruption. You know, when supernovae explode, the luminosity is as bright as the rest of the entire galaxy. For a period of a couple of months, that supernova will be as bright as 200 billion stars. So that means you can see them far away. In fact, astronomers have been able to see type 1a supernova as far away as 9 billion light years. Now, the next slide shows you the light curve of a supernova eruption in our galaxy.
Okay.
So, and this would be for the Andromeda galaxy, any nearby galaxy. The time dilation effect is trivial, and so they're all going to take about 6.5 to 7 months to go through their light cycle. And the light cycle of every type 1a supernova is the same, goes up the same amount of brightness and down. And so a prediction of Big Bang cosmology would be type 1a supernova in galaxies 7, 8, 9 billion light years away, we should see that instead of it being a 6.5-month cycle, it's going to be 8 months or 9 months. And so, you get about a 10 to 15% time dilation effect, but that's measurable.
Right.
I mean, that's going to add about almost a month to the period. So astronomers have accumulated data on literally dozens of type 1a supernova where they see this effect. Now, astronomers would love to be able to test this at great distances, because instead of getting a 10 to 15% effect, you get a 200, 300, 500, 600, 800% difference. It's a spectacular difference. However, we don't have nice clocks like type 1a supernova. And so...
At what point in the universe's history would the stars appear that when they supernova would be in the type 1 category? Because presumably that the reproducibility of the light curve is reflecting the fact that these are the same kinds of stars that are exploding.
Well, we would expect to see not type 1a, but type 2 supernova, which don't have quite as regular a curve, but it's predictable. If you can identify the type of supernova, it has a predicted light curve.
Okay.
Typically much shorter than what you see for a type 1a. So it means it's going to be a little more challenging to measure, say, a 10% difference.
Right.
However, it's far enough away, and the type 2 supernovae are much brighter than type 1a, so you can see them a little farther away. But supernova, you can't see any supernova past about 10 billion light years.
Okay.
We don't have the telescope power to pick them up, although there's some hope that the James Webb Space Telescope is going to be able to see supernova in galaxies, say, 11 billion light years away. But so far, none have been detected.
So the population 3 stars that would be at that distance will supernova, or at least if they're of a certain size, right?
They will, but even the James Webb Space Telescope is going to have a difficulty seeing them, because it's a single star you're looking at.
Yeah.
James Webb can see full-form galaxies at, say, 13.5 billion light years, but they're not going to see single stars. And moreover, supernovae are not that frequent.
Okay.
We get maybe 2 or 3 per big galaxy per year. And so, if you look at millions of galaxies, you're going to see several per year. But that's typically what's happening. We see several per year. We don't see thousands per year.
Yeah.
So astronomers have been saying, okay, we need to come up with another database. And so one that's been used is gamma ray bursts. Because gamma ray bursts, you can see those at 12 billion light years out. And, the burst lasts anywhere from a few milliseconds to, as much as, say 15 minutes, 20 minutes, somewhere in that range.
Do we understand the mechanism now, or do astronomers maybe understand the mechanism of gamma ray bursts?
Yeah, it's one of these really big type 2 supernova eruptions. And you can't see the light, but you can see the gamma rays.
Okay.
Because the gamma rays are compressed into a very short window.
Okay.
A few milliseconds to a few minutes. So you can see these at great distances, whereas you can't see the light of the supernova, because after all, it's extended over a couple of months.
Okay.
And so the energy is spread out. With gamma rays, it's compressed, which is why we can see them at great distances. However, it's not as good a clock as the light curve that you see here.
Right.
And because with the gamma ray bursts, the light curve can be anywhere from 10 milliseconds to, say, about 20 minutes. However, if you've got a really large sample of gamma ray bursts, you can use a statistical study. Okay, statistically, this is what the gamma ray bursts look like that are, say, from 1 billion to 8 billion light years out. So we kind of get a statistical sample, and if there's any kind of time dilation effect, then the sample at, say, 10 to 13 billion years out should show an average variation that's significantly greater. So that's been done. The problem is we don't yet have a big enough sample of cosmic ray bursts, and especially at great distances. I mean, we got a fairly good large sample at nearby distances, not so much at far distances. And so astronomers for the last 20 years have been trying to detect the time dilation, but statistically, they don't have enough of a database to be able to prove it. They can't disprove it. A lot of people say, well, maybe there's a way we can disprove the Big Bang.
Right.
But the data is not there to disprove it, but neither is it there to prove it, although some astronomers think at least we're seeing a hint of the time dilation. Okay, let me take you back, 50 years. It's hard to believe. 50 years ago, I was working on my PhD thesis, and my PhD thesis was quasars in the distant universe. And what I was aware of even back then is that the light curves of these quasars vary. We now know why. The supermassive black holes that are responsible for the quasar emission are accreting matter. And when they accrete a star, there's a burst, which causes the quasar to brighten, and then it dims down. And so you get these fairly rapid variations. By rapid, we're not talking like the gamma rays. We're talking weeks to months of variability. So the time scale is larger, variability over weeks or months. But I remember as a graduate student thinking, okay, it seems like these quasars with certain kinds of flat spectra all have the characteristic of being variable. I mean, that was my PhD thesis, flat spectrum radio sources. And so, and if you got a big sample, there's this average variability. And I speculated, well, the really distant ones, according to Big Bang cosmology, we should see a longer average variability. Problem was, back 50 years ago, our sample size of distant quasars, distant enough where this test could be done, was non-existent. We could see the ones that were up close, we couldn't see the ones far away, and our sample size was in a few dozen. Today, we have a sample size of quasars that's greater than 1 million.
Oh, wow.
So we got a huge database. It's not like the gamma ray bursts where we're looking at a few hundred. We're actually looking at a database of 1 million. Nevertheless, astronomers struggled to see the time dilation effect, mainly because where you can actually measure the variability with some precision is much easier for the nearby ones. But the nearby ones, we're looking at maybe a 10 to 15% time dilation effect. And these are not clocks like supernova, where every supernova has the identical frequency. We have a variability in the frequency.
And when you're dealing with statistical samples, even like a 10% difference could get washed out.
It gets washed out, yeah.
Just in the statistics
The
itself.
statistical noise is bigger than 10%.
Yeah.
So, which meant if we're actually going to see time dilation with quasar variability, we got to look really far away. Because if you look far away, you get a 500% effect as opposed to a 10% effect. So that means that you don't need that big of a sample to actually see, because a factor of 5 is huge. And this paper I just got published in Astronomy and Astrophysics, and it's the first time that astronomers have seen a definitive time dilation effect in the variability of quasars. And what this team of astronomers did is they said, okay, we're going to collect a database of quasars that are variable, that are more distant than 12.8 billion light years, which means we're observing these quasars in the first billion years of the history of the universe, where the time dilation effect is going to be like a factor of 2, 3, 4, 5, or 6. And so they collected a sample. And also, these are quasars that are quite bright, even though they're extremely distant, but bright enough that astronomers have a 2-decade measuring time. So they're actually measuring the variability over a 2-decade period, which means we've got an accurate measure of the time scale of the variability. And they had a sample of 197 quasars, more distant than 12.8 billion light years, which means they had a significant sample. And what they report here is they saw a time dilation effect of an average of 500%,
Wow.
which means it's unmistakable.
Yeah.
There's no way you can say this is a statistical fluke. A factor of 5, it's just so dramatic. And it's exactly what you would predict from a Big Bang creation model where the universe is 13.8 billion years old. So I refer to this as a straightforward and direct test of Big Bang cosmology. There really aren't any assumptions involved here. And another example of the Big Bang creation model spectacularly passing every test that's been thrown at it. I can name 20 tests that astronomers have performed of Big Bang cosmology, but this is one of the most direct tests and one of the most spectacular examples of observations proving, indeed, we live in a Big Bang universe, just like the Bible predicted thousands of years ago. And as a sidebar, it's a dramatic refutation of young Earth creationism, because the only way you're going to get this time dilation effect that we see in the supernova and the quasars is if the universe is about 13.8 billion years old. If the young Earth creationists are correct, then the light curves of these supernova should be a few seconds. They shouldn't be 9 or 10 months. If the universe is infinitely old, they're all going to look to be 7 months. So the fact that we see the ones up close 7 months, the distant ones 8, 9 months, is evidence we live in a universe approximately 14 billion years old. But the quasars really give us a definitive measure that the universe has got to be about 14 billion years old. So that's the bottom line.
Yeah, and again, the idea that the universe has a beginning, obviously Genesis 1:1, Hebrews 11:3.
Yeah, you're not going to get time dilation if the universe has no beginning.
Right.
So, and the kind of time dilation you get depends on the age of the universe. So it tells us the universe can't be trillions of years old, and it can't be thousands of years old. This isn't an accurate measure of the age of the universe,
Right.
but it's a definitive measure that we live in a universe that's approximately 14 billion years old.
Yeah, so the result is consistent with a universe of that age.
Right.
Yeah.
So
Oh, that's
hopefully
cool.
this is a tool that people can use to share with their atheist friends or their young Earth creationist friends,
Yeah.
or anybody, just to say, you know what, the Bible's got predictive power.
Yeah.
It accurately predicts future scientific discoveries.
Yeah, that's cool stuff.
Yeah.
Oh, and you had one more image here I don't think we ended up putting up.
Well, I just wanted to show people what a quasar looks like.
Okay, there we go.
This is the first quasar was ever discovered.
Neat.
Alan Sandage has been here to our office. He's passed away, but he was the one that discovered this quasar, 3C273. It's a quasar that is so bright you can see it through an amateur telescope.
Oh, wow.
So if you got, say, or even if you got a really big pair of binoculars and you're in a dark site, you can see this quasar. And there's a galaxy that surrounds this quasar, but the quasar is so extremely bright that you really can't see the galaxy around it. And that little smoke trail you see out there, that's a relativistic jet that's being pushed out by the quasar. And the quasar is basically a supermassive black hole that's accreting a lot of matter. And as matter comes in towards the black hole, that matter gets converted into energy with about 40% efficiency, which explains why we can see these quasars 12, 13 and a half billion light years away.
Yeah.
Matter of fact, they just spotted a quasar that's 13.5 billion light years. That's a record holder.
Wow.
So James Webb found it.
Yeah.
Yeah.
Neat stuff, Hugh. Thank you. That was a lot of fun hearing that. Okay, well, I'm going to be talking about the computational design of protein machines. And
So we're going from the very big to the very small.
yeah, we are. And, there are these truisms that we hear, right? Like the two that I'm going to talk about today is bigger is better. And then we also hear that bigger isn't always better. So, which is it? And of course, it's really context dependent.
Right.
And so I thought we could have a little bit of fun. I would go through a few examples of of things that are bigger or better if they're bigger, and then a few things that are better if they're smaller. So I'm just going to show you the picture, and let's see if you can guess.
Okay, this sounds like fun.
So these are all things that are better if they're bigger.
Okay.
Can you guess what that is?
Well, I mean, that's a clock. The bigger the clock, the easier it is to see.
Well, it's showing you your account balance.
Okay, yeah. Okay.
So the
The
bigger
bigger
your
my account balance is, the better off I am.
yeah, I
get
think
that.
everybody would agree with that.
Yeah.
Now, here's one you may or may not agree with, but that's
No, I don't agree with that one.
well, but for most people, the bigger the piece of chocolate cake, the better.
Well, if you got a big group that you need to share it with, I go along with that.
Now, here's another one. See if you can. This is a little bit. See if you can guess.
The bigger the flag, or the more stars that are in the flag.
Well, this one is America. In America, bigger is better, right? So most people in the world, when they look at America, they are a little bit befuddled by the fact that everything we do is always very big. Our portion sizes are bigger, our houses are bigger.
Our people are bigger too.
Yeah, right. So in America, bigger is better.
Okay.
This one might be a little nuanced.
Oh, big screen. Yeah.
Yeah, there we go.
Bigger-
Yeah.
Okay.
The bigger the screen, the better for a
Right.
lot of people. This one's a little touchy-feely.
The bigger your heart.
Yeah,
All right.
or love. The bigger
Yeah.
the love is, the bigger your heart. Okay, now here are a few things where smaller is better, right?
Okay.
So,
Debt. Yes.
yeah, right. And then,
The U.S. should pay attention to this one.
yeah, that's right.
Or Congress.
Right. And then here's another one. See if you can guess this.
Okay. A small one handful. A smartphone.
Well, portion sizes.
Portion sizes.
Yeah,
Okay.
so, most people in America want big portion sizes, particularly when you go out to eat. But actually, if you want to live a healthy life, smaller portion sizes are better. This is something I'm beginning to learn to implement in
Well, I
what I eat.
also like a small smartphone because it's easy to transport around.
That's true. Yeah,
Yeah.
that could be something else. Here's something that you and I think would both agree on. Or maybe here it's shorter is better than smaller.
Well, I don't know. I kind of like flying on small planes
Oh,
because they actually give you a little more room for your seat.
okay.
So, and
Well-
you get on and off the plane a lot faster. So, I like small planes.
Yeah. Well, this one I'm thinking flight times.
Flight times.
Yeah.
Okay.
And here's one.
Setting expectations. Yes. The lower your expectations, the less depressed you'll be.
That's right. Yeah. And that's in and of itself depressing, but yes. And then,
Especially on dating sites.
yeah, yeah. Right, right. Okay. And then last but not least is this one
Okay. I'm trying to figure out what we're.
lists.
Oh, lists.
Yeah.
The smaller the to-do list.
Yeah, yeah. The shorter the list,
The shorter the to-do list you get from your wife, the better off you're going to be.
that's right. And then now for something serious.
Okay.
And this is a lot of chemists and material scientists, physicists, engineers are of the opinion that smaller is better. And there's a push among scientists to try to miniaturize systems. And one of those areas where this is the primary focus is nanotechnology.
All fuzz. 67 trillion transistors in this computer you got here.
Yeah.
How do you get 67 trillion transistors unless they're really tiny?
Yep. Yep.
So,
Yeah, well, miniaturization is the thrust, and nanotechnology is the area in kind of science and engineering where the goal is to build really atomic scale machines and
Right.
devices. Some of it involves arrangements of individual atoms. Some of it involves designing molecular systems or supramolecular systems where molecules are interacting. But the goal is to build these devices at the nanoscale. And there are a lot of futurists who think this is going to lead to another industrial revolution. It'll impact industry, agriculture, medicine. So a lot of interest over the last 20 years or so in nanotechnology. And there's a number of approaches that people are taking to try to understand how to build machines at this scale.
You're basically manufacturing molecules, individual molecules.
Right. And then also designing systems where those molecules are interacting with each other to form these stable complexes, yet that have the balance between stability and flexibility so that you can generate motion relative to different components in that system. And so, there's a lot of chemists and material scientists that are just trying to understand basic principles that would allow for nanotechnology. But another approach that people are using is to turn to biochemistry for inspiration. And the reason is because inside the cell is this ensemble of molecular machines that are highly sophisticated, performing complex operations that are also highly integrated.
And they're already manufactured. You don't have to make them.
Right. So scientists and engineers are studying these systems, number one, to just understand basic principles. Two, in some cases, they're co-opting these machines and integrating them into nano devices. And so, biochemistry is really highly inspirational for that reason. And there are two general approaches that are being used. One is to identify biomolecules that have unusual properties that can be exploited in non-natural ways to create nanomachines, and the other is to try to understand these biomachines in and of themselves and use that understanding again to build nano devices. Now, one of the biomolecules that people are using in non-natural ways in nanomachines is DNA. And there are some interesting reasons why DNA is used in this way. DNA's role inside the cell is to store information. And it stores information in the sequence of genetic letters that make up its structure. DNA, I think for most people know this, but just in case people don't who are watching this, consists of two molecular strands where the molecular strands are built from subunit molecules. There are four that are called the nucleotides, abbreviated A, G, C, and T. And those strands then align in a parallel fashion to have a ladder-like architecture where the backbone is the two strands are like the uprights of the ladder. The side groups that interact are like the rungs of the ladder. So that's shown in this particular diagram. But the interaction between the two strands is highly specific, where an adenine subunit is always paired up with a guanine subunit on the opposite strand, and a thymine subunit is always, an adenine unit is always paired up with thymine, guanine is always paired up with cytosine. And so these are called the Watson-Crick base pairing rules. And this actually means that not only will DNA form these highly precise intermolecular interactions between strands, but can form highly precise intra-chain interactions. And that's illustrated in this slide, which is it's a cartoon showing DNA replication at the replication bubble. But notice you have DNA where the two strands are precisely aligned. You've got single strands. You've got places where the same strand is pairing with itself to form these hairpin structures. And because of that, you can actually design DNA sequences to form very complex architectural structures, taking advantage of these properties, the base pairing specificity and the interchain and intrachain interactions. And so this is a whole area of nanotechnology called DNA origami. So the top shows different geometric shapes that can be formed from rods that are interacting with each other. And then the electron micrographs below are actually DNA molecules that somebody has designed that interact with each other to form these very elaborate structures. So you can use DNA to build molecular scaffolding. And in fact, you can even use it to build molecular machines because DNA also has catalytic properties because of the nature of the chemical groups involved.
And because the bonds are already stable, you got a stable structure.
Yes. Right. But you can also get it to dissociate as well. But you get these very precise molecular interactions that allow directed assembly of DNA molecules into these very complex structures. And so that precise molecular recognition is really critical in this process. Now, DNA also has another unusual property. You can actually conduct electrical current along the interior of the double helix, which means people are exploring the use of DNA as a wire to build kind of nanoelectronics. Very interesting stuff. And this diagram is showing a DNA molecule conceptually linking two electrodes together.
Mm-hmm.
And if the DNA double helix is intact, current will flow. If it gets distorted, it disrupts the wire. So it's a very useful.
So how do you solder the two ends?
Well, you chemically attach them.
Okay.
Right, and you probably have quantum tunneling going on, at a molecular scale.
But you're telling me biochemists can actually do the attachments.
Yeah. You can chemically attach the DNA to these electrode materials, and then the quantum, and if there's a polarity between the two electrodes, you're going to get quantum effects which will cause the electrons to hop into the double helix. And then it's actually a quantum tunneling effect that will transport it along the double helix to the other electrode. So pretty cool stuff. But anyway, this is one way in which people are developing nanomachines is, again, to find biomolecules with unusual properties and in a very creative way exploiting those properties to build nano devices in a way that's very different, that's really non-natural compared to, in this case, how DNA is actually functioning inside the cell. Now, the other approach is to look at protein machines inside the cell that are carrying out very complex, elaborate operations and themselves are highly sophisticated machines. With proteins, you get a much greater versatility in terms of how you can combine component parts together to create a much greater array of machines. But here you're now looking at using proteins in the way that they are actually being used inside the cell. So this is really not only direct bioinspiration, but directly a biomimetic effect. And there's a paper I'm going to be talking about in a minute, but this is a quote that comes from the paper where they said, designed dynamic protein mechanical systems are of great interest given the richer functionality of proteins, but with this functionality comes more complex folding and a greater diversity of non-covalent interactions, which despite recent advances in design of static protein nanostructures has made the design of protein machines an outstanding challenge. So in other words, the holy grail of this is to produce nanomachines that are built out of proteins that are novel, that are non-natural machines that are designed by humans. And proteins give you that structural and functional diversity you need to have a very expansive toolkit of options. But with that expansive toolkit of options and that rich functional and structural diversity becomes the headache of not really understanding the principles and what kind of variables you need to manipulate in order to do this. And just as a very quick review, because this will be important as we talk about what these researchers did, here's a very quick overview of protein structure. Proteins are built from amino acids. To first approximation, there are 20 amino acids that are used to build proteins. These all have different chemical and physical properties. And so with the combinatorial possibilities of 20 amino acids, you can create a near infinite number of protein chains. And those protein chains can have a wide range of chemical and physical properties because of those combinatorial possibilities. But then what happens is that chain of amino acids will fold into a three-dimensional structure, and it's a highly orchestrated folding process that's done in a hierarchical manner where the backbone will fold, regions of the backbone will fold into these different conformations. The two most prominent would be alpha helices and beta pleated sheets that are shown there, then those regions of that chain that have different conformations, either alpha helices or beta sheets, will interact in very precise ways to form what are called supersecondary structures or folds. Then those in turn can interact to form domains that interact to form the overall three-dimensional structure of the protein. And then those protein chains that are folded can interact with other protein chains, either ones that are identical to it or different from it, to create complexes.
What you're telling me is that the biochemical engineer faces a challenge of trying to get the folds that he or she wants to see.
Yes.
Because this thing tends to fold on its own.
Right. Now, to appreciate just the type of machine that you can build with proteins, we are going to take a look at a protein called ATP synthase. And this is another quote from the paper where they said, among the best studied and most sophisticated are protein rotary machines as the F1 motor of adenosine triphosphatase, it's also called ATP synthase, or F1F0 ATPase, or the bacterial flagellum, both of which contain axle-like and ring-like symmetrical protein components capable of constrained dynamic motion relative to one another. So these things consist of these protein components that interact, and that interaction now means that those components can move relative to one another around constrained degrees of freedom. But you also have a rigidity to that structure where other motions are prohibited. But it leads to the ability to build a machine like ATP synthase. This is a cartoon, but what you have is this is actually ubiquitous. It's found in every organism on the planet, to my knowledge. It's involved in generating ATP molecules, which are used to power the cell's operations. So because it's ubiquitous means it would have appeared very early in life's history from an evolutionary perspective in LUCA, maybe even in organisms before LUCA. But it consists of a pie, or like a cylindrical type structure embedded in the membrane. That's the motor, where a flow of protons through channels in the motor, essentially its electrical chemical energy, will generate a rotation of a rotor, and that rotor has a cam at the right angle that interacts with the blue and green subunits, causing mechanical movement in a turbine-like action, a pumping-like action, and that mechanical movement drives the generation of a chemical compound, ATP, which is a fuel molecule. But this is converting electrical chemical energy into mechanical energy back into chemical energy. So it's a very sophisticated machine. But what you see there are these hundreds of protein subunits that are interacting to make the F0 component. There's a large number that are interacting to form that rod-like structure that is the rotor, others that are interacting to form the cam. So this is made up of a large number of protein subunits that are interacting to create this structure, which is, again, just phenomenal. And this is a fun quote from Nick Lane. He's an atheist, also a biochemist and an origin of life researcher, and he's written a number of popular level books that are just exceptional books. One of them is The Vital Question. And in the book, he talks about ATP synthase, and this is his statement. It's the most impressive nanomachine of them all. It's a rotary motor in which the flow of protons turns a crankshaft, which in turn rotates a catalytic head. The protein works like a hydroelectric turbine. This is barely poetic license, but a precise description. Yet it is hard to convey the astonishing complexity of this protein motor. This is precision nanoengineering of the highest order, a magical device, and the more we learn about it, the more marvelous it becomes. Some see it as proof for the existence of God. I don't, but it undoubtedly is a wondrous machine. Right? And so, I've used this as an argument that there must be a creator that's responsible for the origin and the design of life because of just the eerie similarity between this machine and the machines that we would build.
Question, Fuz. Has any biochemist been able to duplicate the manufacture of this machine at the net? So it hasn't been done.
And that's going to be part of the point
Okay.
I'm going to make. But no, that's a great question. Now, one way that ATP synthase is kind of like the gold standard. It's the inspiration. By studying this molecule, hopefully principles can emerge that can be applied to build nano machines from proteins. But just for fun, this work is now probably easily 20 years old. A research team looked at actually using ATP synthase as a motor in a nano device. And so this is a proof of principle study where they chemically attached ATP synthase to a machined surface, and then using these protein linkers, they were able to link to the rotor an actin filament, and then by adding ATP or inhibiting ATP hydrolysis, they could get the rotor to rotate or pause its rotation.
So it's like a nano helicopter, right?
Yeah. But the idea is that how do you generate movement in a nano machine? And it's not an easy thing to do because you want that movement to be regulated, to be controlled. And so the idea here is that, well, if we can't build it, why not just take what nature is already providing us?
So they grabbed the motor from a living system, put it on this platform, and then they attached a device to it that it could move.
Right.
That raised another question. You need really sophisticated tweezers to be able to grab one of these things.
Well, it's biochemical techniques where there are techniques to purify the enzyme, and then you, again, would modify it chemically so that it's going to chemically attach. So most of this is just biochemistry, believe it or not.
So they refer to these people as nanosurgeons?
Yeah, something like that.
Okay.
Well, that leads us to this paper that I want to talk about, which is actually published a little bit over a year ago. I end up having all these papers that sit in stacks on my desk.
Oh, you too. I have that same problem.
And then I kind of flip through which would be fun to talk about. And so this isn't cutting edge in the sense that it's about a little bit over a year old, but still it's a remarkable study by scientists from Washington University in Seattle. And they had this idea of building a very crude primitive protein machine from scratch, not in using, again, the inspiration of ATP synthase. And so this is a cartoon on the left of what they conceived their primitive machine to look like. Is essentially going to be an axle and a rotor, where the rotation was going to be driven by Brownian motion. They weren't even looking at how do you create, directed or controlled motion. They're just relying on Brownian motion, where you have an axle and a rotor, a very simple design, and then a couple of end plates that kind of keep it all intact. And then they began to design what the protein's components would look like. And so that's what you see on the right-hand side. So they used essentially this AI-powered computational software to design protein components that would then be ideally suited to assemble on their own into these protein machines. So, for example, and they designed a lot of different variations of this machine. But here's just 3 examples of how they looked to build the rotor. They reasoned, well, if you had an alpha helix that was continuous and formed a circle, that would be an ideal rotor. Of course, you can't make a protein like that in real life. So
so none of these they made from scratch.
No, these are made. They're
all made from scratch.
Yeah. So this is an example of some of the things they designed.
Okay.
So, you can design an alpha helix from first principle. We know enough about which amino acids tend to form alpha helices, which don't, and you can select and choose the amino acids that you want to create different surface structures, different interfacial regions, which is what they were doing.
So were they grabbing groups of molecules or individual molecules?
Right now, this is all being done in situ.
Okay.
or in silico. Sorry, this is all computer modeling.
I get it.
Right.
Okay.
So, but this is essentially laying out their
So this is a blueprint.
exactly. So what they decided to do in the first example is, well, if you could make 3 alpha helices so that the interface between the helices involves highly specific interactions, then once you've synthesized them, they would spontaneously assemble into a ring. And then you could design other protein structures that would interact, at the interface between the different helices and create this rotor-like structure.
Mm-hmm.
By adding an additional alpha helix, you can expand the diameter of that rotor. And then the one on below is they're looking at the interactions of the helices not being end on end, but where one end will interact with the barrel of the alpha helix to create kind of this rotor-like structure. So these are just some examples of how they were going about the design. Here's a couple of examples of how they were looking at developing the rotor. One of them is just getting 3 protein chains that would interact in like a triple alpha helix. And then you could attach to the ends through interfacial interactions these what would you call them? These propeller-like structures made up of 3 different protein subunits. This one on the one on panel B is taking alpha helices and interdigitating them together into a larger rotor where they're interacting kind of in a head-to-head manner or head-to-tail manner. So then once they had the blueprints in place, they then knew what the amino acid sequences were for the different subunits, and they literally synthesized DNA as a synthetic gene that they incorporated into E. coli. And then the E. coli expressed these proteins. They were able to purify them and then get them to assemble in a test tube.
So they got the E. coli to do the manufacturing for them?
Just to make the proteins.
Yeah.
Right. But then they isolated them, and because of the design, they're going to spontaneously assemble into these structures. And so this is showing on the left of each one of these, hypothetically, what that structure should look like if it's properly assembled. And then these are electron micrographs of what they actually were able to assemble in a test tube. So they actually are showing that, in principle, you can start with these computational designs and build these components where you can create variability in the components, and then have them combine in different ways to create. In this case, it's a pretty simple machine that's being powered by Brownian motion. So on one hand, you might say, well, this isn't that big of a deal, but on the other hand, this is a huge milestone in terms of really designing
protein. The reason
protein.
why they're using E. coli is it's the best understood?
Yeah, it's just a very handy, convenient, bacteria that we probably know more about E. coli's biology than human biology.
Right.
And it's used as a workhorse molecule to do this kind of work anyway, where people incorporate foreign genes into E. coli all the time and use it as a bioreactor to produce protein. So, it was...
It's an interesting little factory that we can take advantage of.
Yeah, exactly. But now what was interesting about this work is from the study, they actually were able to develop a set of principles. And what turns out to be key is the interfacial interactions with the different subunits, because you want, again, it to be a highly precise interaction, but you want that interaction to be strong so that it'll drive self-assembly, but you also want to give it enough freedom so that the component parts can move relative to one another if that's what's needed. And so they discovered that symmetry is a very important variable. The complementarity of molecular shapes, which again you have to design, through the computational software that they've developed. Also, the chemical interactions, the hydrophobicity. Can you get electrostatic interactions that will help to stabilize it? Surface area is also very important. And then also the side chain interactions are really critical. So they are understanding now what the variables are, but these have to be precisely fine-tuned in order to get those interactions to be to balance a high affinity and at the same time giving you the degrees of freedom that you need for these machines components to move relative to one another. So it's a pretty remarkable accomplishment that they achieved. Now, what does this have to do with God's existence? Well, we talked about ATP synthase before, and I've written about that in my book, The Cell's Design, and blog articles. It's a very powerful example of the types of machines that you see inside the cell in terms of their elegance and their sophistication. But what interests me the most is how similar the design of ATP synthase is to the designs that we would create as human beings. And we know that that motors that we make are the work of human designers. And when we see machines in the cell that are eerily similar to the machines we would build, it revitalizes Paley's old watchmaker argument. Now, the chief criticism against the watchmaker argument would be, well, evolution can produce these kinds of systems, right? That these are just that evolution has this capacity over vast periods of time to evolve these sophisticated systems. Well, because ATP synthase is, number one, universal, there's not a lot of time for this machine to evolve, because we now have very good evidence that Luca was probably on Earth even at around 3.8 billion years ago, maybe even earlier than that. And so there's not a lot of time for this to evolve. And this is, again, one of the most sophisticated protein machines that we know.
You're going from a period of time when Earth is very hostile to life chemistry to where now you got liquid water, you got solid rocks. But the moment you got liquid water and solid rocks, you got life.
Right. And it's sophisticated at the outset.
And with ATP.
With the ATP synthase, not just ATP, but the synthase, the machine that makes it. But secondly, the second challenge is, but look at what it took these scientists to create very crude, protein motors that on one hand would be heralded as science at its very best, and on the other hand, is laughable compared to the sophistication of ATP synthase. But to design this, they first of all relied on really decades and decades and decades of work of scientists that have come before them that have developed an understanding of biochemical principles sufficient that they even began to know how to develop computer software to go in then and do the computational work needed to design these. They were not just sitting down at a blackboard saying these are the subunits that we need and this is the amino acid sequences. They were using AI-powered, computational systems that are probably being fed training sets of different types of protein structures that are then being used to then direct the design of these, protein components.
It's not designed from scratch. It's basically where they're copying designs they already know about,
right,
and to determine how they should go about making a design.
But the designs are so sophisticated that you need AI technology to even know how to copy them, right? Right. And then you're producing these components that have to have these highly precise, highly fine-tuned interactions, and they've identified maybe a handful of variables that are critical. There's probably a lot more. The more sophisticated these machines are going to be, that they design, the more the variables are going to come into play, right? And so when you look at the amount of intellectual effort that it took and knowledge it took and the use of AI technology to design these things, it really begs the question, is an unguided evolutionary process going to assemble something that is orders and orders of magnitude more sophisticated than what they've been able to produce in something like ATP synthase? So to me, this really undermines an evolutionary explanation, or to maybe phrase it more positively, if this is the amount of intellectual effort it takes to design very crude protein machines, clearly there's got to be a superior intellect that's responsible for biochemistry.
Basically an argument there has to be a mind behind life.
Yes.
A mindless mechanisms aren't going to produce these results.
Right.
And we need a mind that's better funded than the minds that wrote this paper.
Right.
Or with better technology.
Right. Well, and this is exciting stuff because, obviously one of the next steps is how do you generate, coordinated directed motion in these machines at will. But also they're looking at designing other protein components so that you have a library of components eventually that you can combine and recombine, kind of like Legos, to build different types of nanomachines. So it's very exciting work that's going to have huge implications in terms of advancing nanotechnology, but the theological implications are through the roof too, right? When you look at this kind of work.
And only in the 21st century have we had the combination of intellectual talent, technology, and funding to make this even possible.
Yeah, yeah. So smaller is actually better, right? But bigger is better when it comes to the funding of a project like this. So.
It sounds like smarter is better too.
Yes, it is, right. All right, anyway, so that's all I've got for this. So
That's incredible, Fuz.
Yeah. Well, this was a fun episode. Hopefully people have enjoyed it. Thank you so much for watching Star Cells and God today. And we would love to hear your comments. Did you like what you saw? What could we do better? Do you agree with our conclusions? Do you challenge what we're saying? Please let us hear from you. Also, remember to go to our website, reasons.org. Check out the resources we have there. Follow us on social media. And go to our YouTube channel and subscribe. Reasons to Believe is the channel. And then also use the notification button so that you are alerted when the next episode of Star Cells and God drops, which is every Wednesday. And also, Star Cells and God is available on your favorite podcast app. So remember, the more we know about science, the more that we have reasons to believe. Until next time.